Hello, guys. You're welcome to Alicia's MedHub. Today we'll be talking about. Today we'll be doing histopathology, and we'll be. This is an introductory video to pathology. We'll be discussing cell injury and cell death. Now, the objective of this lecture include to demonstrate an understanding of overview of pathology as a course, to demonstrate the forms of cellular adaptation, cell injury, and cell death, to differentiate between necrosis and apoptosis, and lastly, to differentiate between metastatic and dystrophic calcification. Now, definition of pathology. Pathology is a study of structural, biochemical, functional changes which occur in diseases. I'll take it again. Pathology is the study of structural, biochemical, functional changes which occur in diseases. Now, there are several branches to pathology. Pathology is divided into, it has four main branches, and these branches include anatomical pathology, chemical pathology, hematology, and medical microbiology. Now, there are several divisions to anatomical pathology. I say several, there are just main two divisions to anatomical or histopathology. You could either call anatomical pathology histopathology. Now, anatomical pathology has two divisions: general pathology and systemic pathology. Now, general pathology has to do with the basic principles of pathology, has to do with the study of the basic principle of pathology, and these include inflammation, repair, adaptation, cell injury, etc. While systemic pathology has to do with the study of diseases that are specific to a particular system. It is the study of diseases that are specific or associated to a specific system. So now, in the study of diseases, there are four aspects of it, and this include uh, it includes the etiology, which has to do with the cause of the disease and its association, the pathogenesis, which is the mechanism of the disease development, the morphology of the disease, which has to do with structural changes that are associated with the disease, and it could either be gross or histological, right? Or cellular, at the cellular level. Then the clinical manifestation, which has to do with signs, symptoms, signs, and natural cause. So there are certain terms we need to, get their definition right. And we'll start with hemostasis. Now, hemostasis, this is the maintenance of a state of constancy in the body's function and structure. Adaptation, this is a reversible functional and structural response to changes in physiological state or pathological stimuli to achieve an altered state which will enable the cell to survive. Now, cell injury. This is damage to the cells when the limit of adaptive responses are exceeded. Cell death. This is the endpoint of progressive cell injury. Cell death may be normal in embryogenesis or um, hemostasis, right? So they are saying cell death could be a physiological thing, not necessarily a pathological thing. There are certain physiological processes that could lead to cell death and is normal, right? So it's not just associated to a pathological condition where cell die. So it could also be a physiological process. So this is just a diagrammatical representation of um, hemostasis in the body. Now, when a normal cell undergoes stress, right, the cell undergoes adaptive responses. So it undergoes adaptation. Now, this adaptation gives it the ability, um, this adaptation, when the cell lost its ability to adapt, it slide into cell injury, right? Now, the stress, if the stimuli is an injurious one, the cell automatically slide into cell injury. I don't know if you understand. A normal cell, If the stimuli is just stress, the cell adapts, right? It adapts to it. Now, if it cannot adapt, if the cell cannot adapt, cell injury occurs. Now, if the stimuli is an injurious stimuli, straight up the cell will be injured, right? Now, this injured cell, if the stimuli is mild and transient, the injury will be reversed. If the injury is reversed, the cell will become normal. Now, if the stimuli is severe and progressive, there's irreversible cell injury. And when irreversible cell injury occurs, cell death will occur. And cell death occurs in two forms, basically necrosis and apoptosis. Necrosis is the death of a cell, right? While apoptosis is a programmed cell death. Now, hemostasis, which we defined earlier as the maintenance of constancy in the internal milieu, that's in the internal environment, internal system of the body. It requires a balance between physiological demand and constraint imposed on the cell by structural metabolic capacity, differentiation, and specialization. Normal function permits only a minute fluctuations. Adaptation: this is a condition which occurs when physiologic and pathologic stress induce a new steady state in the cell. Adaptation: this is a condition which occurs when physiologic or pathologic stress induce a new steady state in the cell. So both physiological or pathological stress could elicit adaptation in the cell, right? So adaptation allows the cell to survive, essentially, and continue to function in the presence of its stress factor. Now, this stress factor could either be physiological, pathological, whatever it is. The cell adaptation allows the cell to survive. Now, adaptive responses include hypertrophy, hyperplasia, atrophy, and metaplasia. We're going to discuss these things in detail. Now, hypertrophy refers to an increase in the size of cells, resulting in increase in the size of the organ. Define it again. Hypertrophy refers to an increase in the size of cells, resulting in increase in the size of the organ. Now, hypertrophy may be physiologic or pathologic. Example of physiologic hypertrophy include growth of the uterus during pregnancy. Now, during um, pregnancy imposes stress on the that's the female's body. Now, this is physiologic, this is not a pathological process. So, the growth of the uterus during pregnancy is an example of physiologic hypertrophy. Now, there's enlargement of muscle with lifters because of the continuous increase in or continuous increase in the carrying of weight, the the muscle fiber or the muscle cell, they increase in size. Now, example of pathologic hypertrophy include enlargement of the left ventricular wall in hypertensive heart disease because of the increase in the pumping force of the heart. The heart is overworking. The left ventricular wall becomes enlarged, right? Now, so the mechanism for hypertrophy include hypertrophy is induced by increase in physiologic demand, stimulation by hormones, stimulation by growth factors. Hypertrophy is a result of increased production of cellular protein. So, from the definition, hypertrophy refers to an increase in the size of cell resulting in increase in the size of organ. So, the increase in the size of cell occurs when there is increase in the production of cellular protein, right? That's the mechanism of action now. That's what we're talking about. Hypertrophy is a result of increase in the production of cellular protein. So when there is increased proliferation of cellular protein, the cell increases in size. And when there's increase in size, automatically there'll be increase in the organ size. You get the point. So hypertrophy there may also be a change to a more efficient, a may also be a change to a more efficient form of the protein deposited. For example, in cardiac hypertrophy, there is change from the alpha to the beta isoform of myosin heavy chain, which has more energetic contraction. So essentially, it's just explaining um, this point. That's a pathologic hypertrophy when there's enlargement of the left ventricular wall and hypertensive heart disease. So in that process, there is usually a change from the alpha to the beta isoform of myosin heavy chain, which has more energetic you know, contraction ability. Now, hyperplasia is an increase in the number of the cells in an organ or tissue, resulting in increase in the mass of the organ or tissue. Take it again. Hyperplasia is an increase in the number of the cells in an organ or tissue, resulting in increased mass of the organ or tissue. Hyperplasia may be physiologic or pathologic, just like hypertrophy. Now, physiologic hyperplasia include maybe hormonal hyperplasia or compensatory hyperplasia. In hormonal hyperplasia, example includes proliferation of glandular epithelium of of the female breast, as puberty or pregnancy. Um, so what this statement is saying here is there is proliferation of glandular epithelium of the breast. The proliferation of glandular epithelium of the breast it occurs as a result of hormones that are being produced in the female's body. So because of this increase in the production of these hormones, there's increase in the proliferation of glandular epithelium of the female breast and usually occur in puberty and pregnancy. Now, compensatory hyperplasia example is the regrowth of the liver after hepatectomy. Hepatectomy has to do with the resection of the liver. Now, the liver has regenerative capacity to grow. Yes. So, if the liver is resected, there are mechanisms in place, right? There are mechanisms that allows the regrowth of the liver. Now, pathologic hyperplasia may be caused by excess hormone or growth hormone, e.g., endometrial hyperplasia, benign prostatic hyperplasia. Endometrial hyperplasia just there is increased proliferation of um, the endometrium. The endometrial wall is increased in the size of it as a result of excess hormone or excess growth factor. Now, benign prostatic hyperplasia, also known as marginal hyperplasia, there is increase in um, the cells of the prostate gland. Right now, another example could be viral infection, e.g., papilloma virus is causing skin warts, which there is hyperplasia of the epithelium. Now, another important concept is atrophy. Atrophy is the reduced size of an organ or tissue resulting from a decrease in the cell size and number. Atrophy is the reduced size of an organ or tissue resulting from a decrease in the cell size and number. Atrophy may be physiological or pathological. And hypertrophy. Now, atrophy, yeah. So, example of physiologic atrophy include the regression of thyroglosal ducts and notochord during fetal development. During the embryonal development of the thyroid gland, there is a duct that is formed, the thyroid duct. Now, the regression of the thyroid duct and the notochord during fetal development is an example of atrophy. Another example is the reduction in the size of the uterus after giving birth. Now, after a female has put to bed, after a reproductive female has put to bed, the the uterus shrinks back to its normal size. So that process is atrophy. That process is atrophy. Now, pathologic example of pathologic atrophy: decrease in workload, or yeah, causes of pathologic atrophy: decrease in workload, atrophy of disuse when a weightlifter you know, decreases his workload, decreases the weight he takes, he carries, and all of that. There is atrophy of tissues because he's not continuously exercising his muscle. The muscle will shrink back. That's what he's saying. Then loss of innervation. When there's denervation, when a nerve to a particular muscle is severed or disease or injured, it can result into atrophy. Diminished blood supply. Yes, when the blood supply is severed, when there's decreased blood flow to a particular organ or tissue, it can result into atrophy. Inadequate nutrition, loss of endocrine stimulation, pressure atrophy. So all of these things can cause pathologic atrophy. Now there are two mechanisms of atrophy. Number one is atrophy results from decrease in protein synthesis and increased protein degradation. Atrophy results from decreased protein synthesis and increased protein degradation. Degradation occur by ubiquitin proteasome pathway. Now, what this the first mechanism is saying that atrophy occur when there is decrease in protein synthesis and there's increase either that or there's increased protein degradation. There's decrease in protein synthesis or there's increase in the destruction of those proteins that are being produced. Now, and the pathway that is involved is ubiquitin proteasome pathway. And the second mechanism is autophagy. Cell eats itself to survive. Cell components fuse with lysosome and are digested. This reduces cell to manageable size. Now, another very important concept in cell adaptation is metaplasia. Metaplasia is an adaptive substitution of cells in which one differentiated cell type is replaced by another adult cell type. Now, example include number one, ciliated columnar to squamous in the trachea and bronchi of smokers. That is, the normal epithelial lining of the trachea and the bronchi, normal in a normal human being, is ciliated columnar, right? Now, but because of repeated or continuous smoking, the ciliated columnar epithelium is replaced by squamous epithelium. That's an example of metaplasia. Another example is squamous to columnar in Barrett's esophagus. This usually occurs when there is acidic reflux esophagitis. So because of the introduction of a strange chemical to the esophagus, usually the the acid content of the stomach is supposed to remain in the stomach. So because of esophageal reflux, the acid content is refluxing, is going to the esophagus. That exposure of the esophagus to that acidic content causes the replacement of the squamous epithelial lining of the esophagus to columnar, right, which is not supposed to be so. Myositis ossificans, which is the formation of bone in muscle. Now, metaplasia, mechanism for metaplasia, cytokines, growth factors, components of the extracellular matrix around the cell stimulate precursor cells present to differentiate towards a particular lineage. Cytokines, let me take it again, cytokines, growth factors, and component of the extracellular matrix. Component of the extracellular matrix. Component of the extracellular matrix around the cell stimulate the precursor cells present to differentiate towards a particular lineage. Now, metaplasia therefore results from reprogramming of stem cells to produce adult cells which can withstand the stress placed on the tissue. I'll take it again. Metaplasia therefore occurs when there is the reprogramming of stem cells to produce adult cells, to produce matured cells, which can withstand the stress placed on the tissue. Now, this stress is usually pathologic, right? It's usually abnormal. Now, cell injury, the cell may not be able to adapt when the stressful stimuli is severe, leading to cell injury. Cell injury may be reversible or irreversible depending on the degree of stress, the degree of stimuli. Now, causes of cell injury. The causes of cell injury include oxygen deprivation, physical agent, chemical agent, infectious agent, immunological reaction, genetic derangement, nutritional imbalance. Now, morphological changes in cell injury. Reversible injury changes include cell swelling and fatty changes. Reversible injury changes include cell swelling and fatty changes. Now, gross organ organ is appeared, enlarged. That's the organ is enlarged. Gross, looking at the organ grossly, the organ usually appears enlarged. Some may be pallor, some may be increase in weight and size. Now, microscopically, there are clear vacuoles in the cytoplasm of the cell. This results in hydropic changes or vacuolar degeneration. Now, ultrastructural changes that occur during cell injury. Number one is the plasma membrane. There's blebbing, blunting. This blebbing is loss of the microvilli, right? Those structure that enables the cell to move. There's loss of it. That's what it means by blebbing. Now there's blunting and loss of microvilli in the plasma membrane. Um, Another ultrastructural changes occurs in the mitochondria, which is the swelling and unforced densities. Endoplasmic reticulum, another area within the cell, ultrastructure, is endoplasmic reticulum. There's dilation, detachment of the polysome. Of course, the nucleus, there's degradation of granular and fibrillar elements. Now, irreversible injury, necrosis. Now, um, the cytoplasm, there's increase in eosinophilia, there's glassy homogeneous appearance, there's myelin figures, the calcification occasionally. Now, the nuclear changes could include there's karyolysis, there's pycnosis, there's karyohexis. Irreversible injury leads to cell death. Now, the two principal types of cell death include necrosis and apoptosis. Others include necroptosis, pyroptosis, ferroptosis, autophagy. Necrosis can be defined. It is defined. Well, let me just put it the way it is in the material. Necrosis is the sum of morphological changes that follow cell death in a living tissue or organ. Please note the definition, it's so important. Necrosis is the sum of morphological changes that follow cell death in a living tissue or organ. Cells in a specimen fixed in formalin are dead but not necrotic, right? So essentially, what this page is saying is necrosis occurs in living tissue, while putrefaction occurs in the dead. So necrosis is the sum of morphological changes that follow cell death. When a cell dies, the changes that follows it still within the living tissue known as necrosis. Now there are several patterns of necrosis, and these patterns include coagulative, liquefactive, gangrenous, caseous, fat necrosis, fibrinous necrosis. Now, the six mechanisms of cell injury. These mechanisms include ATP depletion, mitochondrial damage, increased cytoplasmic calcium, increased reactive oxygen species, plasma and lysosomal membrane damage, protein misfolding, and DNA damage. Now, ATP depletion has two primary outcomes. When there is depletion of oxygen, energy, yeah, that's the right word. When there's depletion of energy to a cell, there are three things that occur. And uh, one of them is rather there's decrease in sodium pump, When there's decrease in sodium pump, it results into increase in influx of calcium into the cell. Now, this will also result in um, increase in influx of water and as well as sodium, ultimately to result in endoplasmic reticular uh, endoplasmic reticulum. It results in the swelling of the endoplasmic reticulum, which will result in cellular swelling. Then loss of microvilli and blebs, formation of blebs. I explained what blebs, loss of those cellular protrusion, loss of it. So the second outcome is there's increase in anaerobic glycolysis, which will result in decrease in glycogen as well as increase in lactic acid and decrease pH. When there's increase in lactic acid production, that's increase in the acidity of the cell, it will result to decrease in pH, right? Which will result in clumping of nuclear chromatin. So the cell dies ultimately. Now, the third outcome of ATP depletion is detachment of ribosome. When there's detachment of ribosome, it will result in decrease in protein synthesis. We all know that ribosome is involved in protein synthesis, ultimately leading to lipid deposition within the cell. So this is just a diagrammatical representation of um, uh, okay, diagrammatic representation of the cause of cell injury. So when there's ischemia, so there's ischemia which will result in um, loss of blood supply to the tissue and organ. So there will be decrease in oxygen supply to the mitochondria. So this will result in oxidative phosphorylation of ATP. When there's decrease in oxidative phosphorylation of ATP, there's decrease in ATP, right? So that's decrease in the formation of ATP. So when there's ATP depletion, right? All those processes, those three main processes, or those three main outcomes that we spoke about here will occur. So now, the first thing, which is there's decrease in sodium pump, which results in increase in influx of calcium, water, as well as sodium. Um, Concurrently, there will be efflux of potassium. So ultimately, this whole process will result in endoplasmic reticulum swelling, cellular swelling, loss of microvilli, and blebs. Now, the second outcome is anaerobic glycolysis, which will result in decrease in glycogen production, right? Which is the glucose storage system of a cell. So ultimately, there will be increase in lactic acid, so resulting in decrease in the pH. When so decrease that is tilting to the more acidic nature. So because of the increase in acidic content of the cell, there will be clumping of the nuclear chromatin. Ultimately, the cell will die. So another outcome is detachment of the ribosome. The detachment of the ribosome, which results in decrease in protein synthesis and as well as increase in lipid deposition. There will be increase in lipid deposition within the cell. Uh, Mitochondrial damage. Mitochondrial damage results in two outcomes. The two outcomes result from mitochondrial damage includes necrosis or apoptosis. Now, the first one, formation of mitochondrial permeability transition pore will result in loss of membrane potential, which will ultimately result in inability to generate ATP. Then necrosis will result, right? So when the mitochondria is damaged, there will be formation of endochondral permeability transition pore. So the the mitochondrial membrane will be perforated. Now, when there's this formation of this transition pore, with loss of membrane potential, the mitochondrial membrane will its potential will be lost. So this will result in inability to generate ATP. That's the mitochondrial function, which is the powerhouse of the cell, production of ATP, will be lost. That function will be lost. Ultimately, necrosis will occur. Now, another process could be the leakage of the cytochrome C, another pro-apoptotic proteins, so which will result in apoptosis. So, this diagram explains everything I've just said. So, just you can go through it yourself. Now, the third process is increased cytosolic calcium. The third process that can lead to cell injury, increased cytosolic calcium. It has three major consequences. Number one is the plasma membrane damage, result from activation of phospholipase and protease. The second is nuclear membrane damage, resulting from activation of endonucleases. Then the third is decreased ATP because of the increased mitochondrial permeability transition. ATP is also activated. This is a diagrammatical representation of what we have just discussed. I Because this material is quite long, so I may just skip some things. Now, so generation of free radicals. Free radicals are chemicals species that have a single unpaired electron in an outer orbit. This makes them unstable and very quickly uh, to react with any adjacent molecule and compounds like protein, carbohydrates, fat, nucleic acid, etc. Now, free radical induced injury occurs in conditions such as chemical and radiation injury, reperfusion injury. Reperfusion injury is a paradoxical exacerbation of cellular injury that occur um, when a previously ischemic tissue is supplied by blood again. When the blood supply to a tissue is cut, when there is restoration, when the blood supply is restored, there are certain processes that can result in injury of the cell. That's just what reperfusion injury means. Then cellular aging. Free radical may be generated by redox reaction during normal metabolism, absorption of radiant energy, UV light, X-ray, during inflammation by activated leukocytes, enzymatic metabolism of exogenous chemicals or drugs, transition metal, e.g. iron, copper, nitric oxide. Removal of free radicals occurs by antioxidants, example vitamin E, vitamin A, ascorbic acid, glutathione, metal binding plasma proteins, e.g. transferrin, ferritin, lactoferrin, ceruloplasmin, enzymes such as catalase, superoxides, dismutase, glutathione peroxidase. Free radical cause damage to cell by three methods. Free radical cause damage to cell by three methods, and these methods include number one is lipid peroxidation of membrane. Membrane lipid are converted to peroxide, also unstable, leading to extensive membrane damage. Secondly, is oxidation modification of protein. Oxidation of amino acid side chain. Protein-protein cross-linkage formation. Oxidation of protein backbone. Thirdly, there's lesion DNA. Single or double-stranded breaks. There's cross-linking of DNA strand. And lastly, there's formation of adduct. So this is a diagrammatic representation of everything I just said here. So you can just follow it. You can just follow it. Now, membrane damage. Four mechanisms of membrane damage. Number one is reactive oxygen species. There's reduced phospholipid synthesis because of ATP depletion. There's increased phospholipid breakdown caused by increased cytoplasmic calcium. The cytoskeletal abnormalities caused also by increased cytosolic calcium. There are three causes of membrane damage. The three consequences are mitochondrial membrane damage, which will result in ATP depletion, the release of apoptotic protein. Secondly, there's plasma membrane damage. There's loss of osmotic balance. It will result in influx of fluids and ion into the cell. There will be loss of cellular content. And thirdly, there will be lysosomal membrane damage. There will be leakage of enzyme into the cytoplasm, which can result into autophagy, if you remember. Then there's ribosomal RNAs, there will be DNAs, there will be protease, there will be phosphatase. So this is also a diagrammatic representation um, of the consequence of membrane damage. So this the six major mechanism of cell injury. The six major mechanisms of cell injury, which is ATP depletion, mitochondrial damage, entry of calcium, that's increased cytosolic calcium, increase in reactive oxygen species, membrane damage, protein misfolding, and DNA damage. So ATP depletion results in multiple downstream effects. If like we stated here, ATP depletion results into this 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 event. So you can go back and look it up. So let's conclude this section by stating that physiologic or pathologic stress, when mild, induces adaptation such as hypertrophy or hyperplasia. Severe stress or toxic exposure leads to cell injury, and this cell injury may be reversible or irreversible, causing cell death, which cell death could be either necrosis or apoptosis, depending on the initiating factor. Now, the point of irreversibility is undefined, right? The point of irreversibility is undefined but identified by mitochondrial membrane damage. So for um, the hallmark or the identifying feature for irreversibility of cell injury is mitochondria. When there's mitochondrial damage or when there's membrane damage, you know that this cell has gone to a point of no return. Whatever is happening to the cell, it cannot be reversed. So, uh, yeah, we looked at this image earlier. Now, apoptosis. So the two forms of cell injury or cell death, sorry, the two forms of cell death are apoptosis and necrosis. So we have to we'll discuss them. We'll discuss them now. So apoptosis simply means genetically programmed cell death. Apoptosis and necrosis may occur concurrently, right? Mitochondrial membrane damage. Apoptosis may be physiological or pathologic. Physiologic apoptosis examples of physiological apoptosis include programmed destruction of cell during embryogenesis, evolution of hormone-dependent tissue upon hormone withdrawal, e.g., endometrium in menstrual cycle. This is self-explanatory, right? When during the ov- during the menstrual period of a female, a reproductive female, when that cycle fades away and there is withdrawal or reduction in the hormone within the bloodstream, the endometrium shrinks back to its normal size, right? So that's just what it's saying. Then there's cell loss in proliferation, population, populations, immature lymphocytes in bone marrow. Thirdly, there's cell loss in proliferating cell populations, immature lymphocytes in bone marrow. Fourthly, there's elimination of potentially harmful self-reactive lymphocytes, removal of neutrophils and lymphocytes at the end of immune response or inflammation. All these are physiologic apoptosis, right? So take note, all these are physiologic apoptosis. Now, pathologic apoptosis include DNA damage, accumulation of misfolded proteins, which will result in endoplasmic reticulum stress induced by excess protein in the endoplasmic reticulum, responsible for neurodegenerative diseases. But thirdly, there's viral infections induced by cytotoxic T cells. And fourthly, pathologic atrophy in parenchymal organs after duct destruction. Now, morphological changes that are associated with apoptosis include cell shrinkage, chromatin condensation, formation of cytoplasmic bleb and apoptotic bodies. I explained cytoplasmic bleb, phagocytosis of apoptotic cells and bodies by macrophages. Apoptotic cell appears as a round or oval mass with extensive eosinophilic cytoplasm with fragments of dense nuclear chromatin. So it's essentially this statement is just describing a cell that has undergone apoptosis. Now you know a cell that has undergone apoptosis, um, by its appearance. It appears as a round or oval mass with extensively eosinophilic cytoplasm. So there's eosinophil within the cytoplasm with fragments of dense nuclear chromatin. So this is an apoptotic cell. An apoptotic cell. Now the mechanism of apoptosis. The process of apoptosis is divided into two phases. We have the initiation phase, we have the execution phase. We have the initiation phase, we have the execution phase. Now, initiation occurs by two pathways. Intrinsic, also the intrinsic pathway, also known as the mitochondrial pathway, and the extrinsic pathway or the death receptor pathway. Um, the intrinsic pathway of apoptosis, this is started by the release of cytochrome C from the mitochondria into the cytoplasm. The release of cytochrome C is controlled by the balance between pro and anti-apoptotic membrane of the BCL family of proteins. Anti-apoptotic gene include BCL2, BCLX, MCL1, while pro-apoptotic gene includes the BAX and BAK. Regulated apoptosis initiator include BIM, BID, BAD, PUMA as PUMA, and NOSA. Being bead or bad initiates back when the cells are deprived of survival signals. DNA damaged, misfolded protein induce endoplasmic reticulum stress. Being bead or bad activate backs and back when the cells are deprived of survival signals, DNA damage. Anyway, what this thing is saying is the beam bead bad, right? They are regulated. They regulate apoptosis initiator, right? They regulate apoptosis initiator. Now, when a cell is deprived of survival signal, DNA damage and misfolded protein induce the plasmid stress. Now, when a cell is undergoing any of these processes, right, the beam bead or bad, which are apoptotic, they regulate the process of apoptosis initiator. When the cell is undergoing any of this gene activates the backs and back. And what are the backs and back? The backs and back are the pro-apoptotic. That means they promote apoptotic process, right? So when a cell is undergoing any of this apoptotic initiator, or they regulate the process of the initiation of apoptosis, they will activate these backs and back, which are pro-apoptotic gene, right? So that the cell will undergo apoptosis. Yes. So the back and back form oligomers which insert into the mitochondrial membrane and create channels which allow protein cytochrome C from the inner mitochondrial membrane to leak out. They also block the function of anti-apoptotic proteins which are BCL2 and BCLX. It's self-explanatory. Self-explanatory. So cytochrome C, or let me just explain it. So apart from the fact that the beam bead or bad activates the pro-apoptotic gene, they also inactivate, they also block or inactivate the activity of anti-apoptotic gene, which are the BCL2 and BCLX. The anti-apoptotic inhibits the process of apoptosis, right? So the beam bead bad, which are apoptotic, which are the genes that are responsible or that regulate the process of apoptosis, also block anti-apoptotic gene. right? So to prevent, the stoppage of apoptosis, right? So it's quite simple. So cytochrome C then binds to um, APAF1, that is apoptosis activating factor, APAF1. Yeah. So this complex then binds to caspase 9, right? This is the initiator caspase for intrinsic pathway, and the cell then proceeds into execution phase. Now, what this place is saying is that cytochrome C, when it leaks out of the mitochondria, it binds to APAF1. APAF1 is known as apoptosis activating factor. Now, the the interaction between cytochrome C and APAF1, the complex that is formed then binds to caspase 9. This this interaction ultimately is the initiator caspase for the caspase 9 is the initiator caspase for intrinsic pathway. Intrinsic pathway. And the cell then proceeds into execution phase. So this is a diagrammatic representation of what we have spoken, right? So I may not go into the details of this diagram because of time. I don't want this video to be too long. Yeah. So the extrinsic or the death receptor, uh, yeah, the extrinsic pathway or the death receptor initiated pathway. This involves the use of death receptors present on the plasma membrane of many cells. Death receptors are membrane of the um, TNF, that's small necrotic factor family, which have cytoplasmic depth domain. Best known is the type 1 TNF receptor with related protein FAS CD95. The ligand which binds to the FAS is expressed on many T cells that recognize self-antigen and cytotoxic T lymphocytes. FAS ligands, FAS-L binds to FAS. FAS then binds to FAD. FAD is an abbreviation for fast-associated domains. FAD is an adapter protein which also has a death domain. FAD, which is um, um, fast-associated death domain, then activate procaspase 8 to form active to catalytic caspase 8. Caspase 10 is the form of this caspase found in humans. Caspase 10 is the form of this caspase found in humans. So instead of caspase 8, we have caspase 10 in humans. This is the initiator caspase for the extrinsic pathway. So the initiator caspase for the extrinsic pathway is caspase 10, right? That's in humans. Um, I don't know whether in lower animals is caspase 8, but in humans caspase 10 is the form of this caspase found in humans. So again, if you look at the image, both intrinsic and extrinsic pathway converge on the executional pathway. Caspase 9 from intrinsic pathway and caspase 8 or caspase 10 in humans, caspase 8 from the extrinsic pathway. Each activate the caspase 3. So caspase 8 is coming this way, caspase from the intrinsic pathway and caspase 8 or caspase 10 is coming from the extrinsic pathway. Altogether activate the caspase 3 or caspase 6, which are the executional caspases, right? So these things, you need to know these things down so that for the sake of MCQ, if your school writes MCQs, it's important you note these things down, right? So some action of exogenous caspases include number one, cleave cytoskeletal and nuclear matrix protein. They disrupt the cytoskeleton of a cell. They cause nuclear breakdown. They cause the cleave protein involved in transcription, DNA replication, and DNA repair. Now, phosphatidylserine are exposed on the apoptotic cells and are recognized by receptors on the surface of the macrophages. Some apoptotic cells secrete thrombospodin recognized by macrophages and phagocytes. Apoptotic bodies may be coated by complement protein. An exin 5 stain is used to identify apoptotic stain. This is a very important MCQ question. An example of a stain that can identify an apoptotic cell. is annexin V stain, annexin V or annexin 5 stain, right? So disorders of apoptosis, disorders associated with defective apoptosis include uh, defective apoptosis leads to increase in cell survival, right? When there is a defect in the process of apoptosis, the life of the cell increases. So it increases the cell survival. It may lead to survival of abnormal cell. Very important. You know, during the process of cell development, maturation, right, there are certain cells that may have genetic aberrations that are not supposed to continue in that line because if they do, they could result into the formation of production of cancer cells or defective cells. So the body has a way of channeling these cells, or the body has a way of initiating apoptotic processes. Now, when these apoptotic processes fail, it may lead to the survival of these abnormal cells, right? It may lead to the survival of abnormal cells. So, survival of cells with mutation, e.g., mutation in the P53, may lead to cancer. Yes. Now, the P53 is, I will make a video about P53 gene. But for now, just know that the P53 gene is a cell that is involved in the is a policeman, right? it it ensures that any abnormal cell, undergoes apoptosis dies. Now, when there is a mutation in this P53 gene, any cell, and apoptotic process is defective, that means that cell will, its survival rate will increase. And if its survival rate increase, it may lead to cancer cell production of cancer cells, and we don't want that, right? So also, it could lead to defective apoptosis may be the basis of autoimmune disease with failure to eliminate self-reactive lymphocytes. So it's what the statement is saying that defective when the process of apoptosis is defective, it may result. It can. Yeah, there's a possibility that it may be the basis for autoimmune diseases, where the immune system of an individual is attacking the body, right? So that's that. Disorder associated with excessive apoptosis. That's when apoptotic processes are not regulated. It may lead to excess cell death. Example include neurodegenerative diseases, ischemic injury, um, myocardial infarction, stroke, death of viruses infected cell. Now, pathology classifications. Pathology classification is the abnormal tissue deposition of calcium salts and small amounts of iron, magnesium, and other salts. So, pathology classification is the abnormal deposition of calcium salt and small amount of iron, magnesium, and other tissue, another salt in tissues. There are two forms of pathologic classifications recognized. We have dystrophic classification and metastatic classification. Dystrophic classification occurs in areas of necrosis. Please note this. Dystrophic classification occurs in areas of necrosis. It appears grossly as gritty deposits. Microscopically, it appears as basophilic amorphous granular, sometimes clumped appearance. Heterotopic bone may form. Samoma bodies in some papillary cancers is an example of dystrophic calcification. Asbestos bodies appearing beaded in the lungs. So the Samoma bodies in an ovarian cancer is is um, is is an indication of dystrophic calcification. That's abnormal deposition of calcium salt within the tissue. Now, pathogenesis of dystrophic calcification. Deposition ultimately involved the precipitation of a crystalline calcium phosphate similar to bone hydroxyapatite. Two phases: initiation and propagation. Now, the initiation, extracellular or intracellular. Yeah, initiation involves extracellular and intracellular. Now, extracellular occurs on the membrane-bound vesicles containing charged phospholipids. Phosphate formed binds to calcium. Now, intracellular calcium ion is deposited in the mitochondria of dead or dying cells. Now, for the propagation of crystal formation depends on the constitution of calcium and phosphate, the presence of inhibitors, and the structural components of extracellular matrix. Metastatic calcification. Metastatic calcification is abnormal deposition of calcium in normal tissue when there is hypercalcemia. So hypercalcemia simply means an increase or an elevated amount of calcium in the blood. When there's when this occur, it will result in the abnormal deposition of calcium in normal tissues. So where calciums are not supposed to be found, they'll be found there, right? So there are four principal causes. Number one is increased secretion of parathyroid hormone, and you understand when there's increased secretion of parathyroid hormone, it will stimulate. There'll be increase in resorption of calcium from the GI, decrease in excretion of calcium, so ultimately decrease in bone resorption, right? So ultimately it will result into increase in the calcium level in the blood, so resulting in abnormal deposition of calcium in normal tissue. Now, bone destruction, e.g., multiple myeloma, bone metastasis, immobilization, Paget disease. Thirdly, vitamin D related disorder, e.g., vitamin D intoxication, psychosis, Williams syndrome, renal failure, which will result in retention of phosphate, ultimately resulting in secondary hyperparathyroidism. Now, cellular aging, the functional and structural changes in aging are influenced by genetic factor. The functional and structural changes in aging are influenced by genetic factor, diet, social condition, and the presence of age-related diseases such as diabetes mellitus, atherosclerosis, and osteoarthritis. Cellular aging is a result of steady decline in cellular function and viability caused by genetic abnormalities and the accumulation of cellular and molecular damage due to the effect of exposure to exogenous influences. Factors which contribute to aging. The factors which contribute to aging include cellular sequences, environmental insults, DNA repair defects, abnormal growth factor signaling. Cellular sequencing. This refers to the concept that cells have a limited capacity for replication. Telomere shortening is a mechanism of cellular sequencing. Telomere, short repetitive sequence of DNA at the end of chromosomes. Each cell replication shortens the sequence. Stem cells and germ cells maintain their telomere length by using telomerase enzyme. Somatic cell lack telomerase, so a critical stage is reached when the cell can no longer divide. Accumulated, accumulated metabolic and genetic damage. Reactive oxygen species induce damage. Now, when there's DNA damage, when the DNA damage is not repaired, it will result in defective DNA helicases. Werner syndrome, which is premature aging syndrome. There's accumulation of damage organelles. Calorie restriction has been found to prolong life in animals. Interesting. Caloric restriction has been found to prolong life in animals. So anyway, this is just a diagrammatical representation um, of what I have spoken here. So um, thank you so much for listening. If you found this video helpful, please kindly like and subscribe. Thank you so much.